biodiversity. Jessica's talking about variability and connectivity in giant and bluefin kingfish. Thank you, Jessica.
Can you all hear me okay? Kind of tall. Great. Cool. Yeah, we're going to switch gears a little bit and talk about genetics. And if you run away, I'm going to put some genetics questions in quiz night tonight. So just kidding. Yeah, so I'm Jessica Glass. I'm in the fisheries department at the University of Alaska Fairbanks. I'm going to start my timer. And I'm a research associate at the South African Institute for Aquatic Biodiversity, also known as SIAB. So my lab studies how evolution can inform the management and conservation of fisheries across the Indo-Pacific. And we do that by looking at the intersection of genotype or DNA, phenotype, physical traits, behavior, and environment. And for the last several years, I focused on this clade of fishes. Luckily, with this audience, these should all look familiar to you. And primarily, I've been focusing on the carangoids, or the Carangidae family within the carangoid clade, which is the most diverse family within this clade, and that consists of the scads, the trevallies, the jacks, kingfish, lots of different names for those species. And of course, we are here at this conference because a lot of species of the trevallies are iconic sport fishes due to their habitat preferences, their size. They put up a big fight, both in the fly fishing industry and the rod and reel fishing industry. These are immensely popular fish. And particularly, I think there's an animation here. The bluefin trevally and the GT, or giant trevally, or giant kingfish, have a lot of cultural importance across their range as well. So in island communities like Hawaii, Tahiti, Samoa, there's a lot of traditional folklore, traditional fishing methods, ways of catching these species. There's according to FishBase, over 200 colloquial names for the giant trevally across the world. And so these are incredibly important species, not just for the recreational fishing industry, but for communities and livelihoods. And it's the access rights and fishing rights in many of these communities are being challenged in the face of resource allocation and potential overfishing. But there's some really basic biological information that we still don't know about these species, which is incredibly important because across most of their ranges, they remain mostly unmonitored and unmanaged. And so they have pretty wide ranges. You can see here the bluefin range is in blue and the GT range is in red, and the areas where they overlap, which is most of their range, is in purple. They have this large Indo-Pacific range. And so with that big range, we can ask some of these big questions and use big data to answer those. And that was sort of the aim of this series of work. And I'm kind of today presenting a compilation of many different studies that we've done. So one big question, right, is what's the extent of connectivity across the Indo-Pacific given these large ranges and given what we know about like movement behavior so far from these species? They're not big pelagic tunas that are migrating, across the open ocean. So the first question that we were interested in was looking at genetic or demographic connectivity. So are there distinct genetic populations? Are there biogeographic barriers that are known? And when I started this work, the only genetic study that had been done at the time was in Hawaii, which is of course a very small proportion of their range. And then leading into that, we were really interested in trophic or ecological connectivity. Is there niche partitioning happening across populations? And is that due to, for example, differences in habitat type? And then lastly, you can think about actual physical connectivity and movement behavior. And that's work that colleagues of mine have done using acoustic tagging. Now, really big range, PhD student budget, only one person, right, or as part of a small team. You can't be everywhere at once, right? And so this work relied a lot on collaboration with recreational anglers, with sport fishers across 13 different localities in Indo-Pacific. This ended up being a really exciting and fruitful component. And through the networks of collaborators charter fishing operators and anglers from all over the world ended up reaching out and saying, how can we help? How can we help? We're hearing that you're doing this GT work. We want to help. So we were able to send and create these little sampling kits, secure the permits, and it really took off. And some of the anglers got really excited about it. They posted on social media. Some of the charter operators hired professional photographers to come and take pictures of their clients collecting the fin clips, for example. So this was a really, not just like cool side component, right, but like a really crucial and successful part of this work. That collaboration with charter operators and sport fishers really took off in Seychelles, where particularly in the atolls and coralline islands, these fish are really important. They're really heavily targeted by the recreational fishing industry. So for example, this is the island of Farquhar, and 100% of fly fishing guides rank trevallies as their #1 most targeted species on that island. And we know that from work that was done by my former master's student, Sheena Talma, who is from Seychelles and was the first person to start conducting a socioeconomic survey of the recreational angling fishery, fly fishing industry across the atolls and islands of Seychelles. And because building on Sheena's work, we were able to talk to many of these operators and see, like, what are their concerns? What are your priorities? How can our research help potentially answer some of those questions? And I'll say there's still a lot of questions that remain unanswered, but that's the fun of this, right? Another component of this was building collaborations with scientists who were studying, like I mentioned, the movement behavior and the physical connectivity of these animals. So Paul Cowley, Ryan Daly, and we're tagging these fish, they still are tagging these fish in Southern Africa and Seychelles and understanding their connectivity patterns. I'm not going to speak to that. Ryan's giving a talk on GT movement behavior on Thursday. But I want to make the point that as they were going and tagging these fish, we were able to get fin clips for the genetic analysis and muscle tissue samples for the stable isotope analysis. So within this framework of genotype, phenotype, behavior, and ecology, we could apply those approaches and answer a lot of those questions just from a single sample, single fish. So as I mentioned, we had samples collected from 13 localities across the Indo-Pacific. This is just a subsample after data filtering for the genomic analysis. A lot of those came, like I said, from recreational anglers, but some also came from academic institutions and museums and so on. And so getting into the results, at the same time this was building across the world with these angling collaborators, I formed a new collaboration with scientists at BYU who in the last couple of years worked with me to publish the first whole genome annotation of GTs and bluefins. And those are publicly available. It's an amazing resource for anyone that might want to be going through and looking at genetic studies or transcriptomic studies or epigenetic studies, anything that's related to the omics of these fish. So we were able to map RAD-seq data across the genome, and I'm not going to go through the details of the methods, but we were able to generate tens of thousands of SNPs or mutation sites that we were able to then use to look at the genetic connectivity and structure of these populations. And what we found was quite interesting. So in both cases, so these are PCAs, principal component analysis plots, where points are individual fish and points that are clustered more closely together tend to be more genetically similar. And what stands out for the bluefins is that along PC1, the major kind of separation here is this separate cluster of individuals from Hawaii. And then we see a little bit of separation between the West Pacific and the Indian Ocean. We see something similar with the giant trevallies, but also inclusive of the island of Kiribati, of Christmas Island. So there's some separation there. And then we have a West Pacific Eastern Indian population, and then points that are very tightly clustered, very genetically similar in the Western Indian Ocean. If we look at that another way, this is a modeling approach. So these are individual, got kind of cut off there, whatever. Individual ancestry plots showing putative populations and estimated proportion of ancestry. So every bar here is an individual fish, and this is for bluefins. And as you can see, that this bottom one is 3 putative populations, the top is 2. We see this evidence of a separate genetically distinct population in Hawaii with potentially some one-way gene flow primarily from the Central and Pacific and Eastern Indian Ocean, or sorry, Western Pacific and Eastern Indian Ocean into the Hawaii, which is a common trend. Things kind of end up in Hawaii, but it's harder for them to leave due to the ocean currents. Okay, similar for the GTs, we see this evidence of a separate putative genetic population in Hawaii and Kiribati, and some evidence actually for a third population that would be sympatric, overlapping in range as you go from east to west. Got to get my orientation straight here. From the Central Pacific into the Western Indian Ocean. Another way to look at that is through a heat map of pairwise genetic differentiation, or FST. And here again, you can see that we find strong signal of differentiation, so higher FST values when we compare Hawaii and Kiribati compared to the rest of the population, and then this sort of gradient of genetic differentiation as we move from east to west, and then very similar genetic structure in the Western Indian Ocean. So those are all very closely related, not a lot of allelic differentiation between those populations. So on that note, right, we find a lot of admixture in the Western Indian Ocean. I'm skipping ahead of myself here. Another thing to point out too is we have those separate populations genetically in Hawaii and Kiribati, and we find that there's significantly reduced genetic diversity at these locations, which is really important. We measure this in terms of heterozygosity. We think about the importance of genetic diversity in sustaining populations in the face of disease or overfishing or external or internal forces of evolution that might inhibit the success of that population. And in Hawaii, giant trevallies are overfished, and overfishing is still occurring according to the last NOAA estimate. This is also concerning because this is a federal agency, and most of the fishing for these species occurs in state waters where there's not consistent estimates of abundance or fishing monitoring. So understanding kind of the implications there from the genetic study is really important in saying, okay, this is a really special population. These fish are unique to this region, and the population might be at risk, right, because of that low genetic diversity. So what do we do with that is the question. Okay, back to what I was saying before, Western Indian Ocean has a lot of genetic connectivity. So we were then interested in the trophic connectivity and the ecological connectivity. And so we looked at stable isotope data from 4 different sites that represented different habitat types in Seychelles. We have Mahe Island, which is the granitic island. We have 2 atolls in Seychelles. And then we have this coastal reef site in Mozambique that was a reef that's about 4 kilometers offshore. And we were really interested in using trophic position as a proxy for diet. This is nice when you're thinking about catch and release fisheries because you don't have to take stomach contents, so you can just take a muscle tissue sample and generate estimates of carbon and nitrogen isotope ratios. We used compound-specific amino isotope analysis. If you're in the isotope world, then you know what that means. If not, happy to talk about it afterwards. It just means basically that we don't need a baseline for nitrogen. And we were really interested in this, not just trophic position, but also environmental signature, given these different habitat types. And the first thing that we found that is maybe unsurprising, but giant trevallies, giant kingfish are top predators, right? They have the same trophic positions in some of these sites as bluefin tuna, even pushing close to great white sharks, blacktip sharks, and other shark species. We also see this sort of a signature of carbon from an offshore signature at these sites in Mozambique and the granitic islands of Mahe, and then more of a coastal signature as we move into some of these atolls. So there is, trophic niche partitioning occurring that seems to be correlated with habitat type. We also found signatures of ontogenetic changes, so different signatures in juveniles versus adults. I'm only going to show the adult information here, but what I want to point out is that contrary to what you might expect in some of these large fishes where you expect a higher trophic position as they get larger, in some cases we didn't see that, right? So in some samples from St. Joseph and Mahe, some of the biggest fish actually had some of the lower trophic position estimates that we generated. Thanks. Yeah. And lo and behold, Ryan and colleagues actually tagged some of these fish in St. Joseph Atoll. And what we found was that two of these individuals were going to this other island about 70 kilometers away. And not, of course, not knowing their stomach content analysis, we can't tell what they were eating, but it could be suggestive of potentially individual specialization in diet for these fish. Okay, so to wrap up, I hope that I showed you that with this kind of crux of sport fisher and recreational fisher collaboration, we were able to look at this intersection of genotype, phenotype, behavior, and environment. And look at all these different facets of the biology of these species from these collaborations, and just using a single sample to answer all of these questions, single fish. So it's really exciting. There's a lot more questions at hand. In the future, we're looking towards filling in the gaps, right? So really, really narrowing down where are those biogeographic breaks. Also looking at the prevalence of hybridization between bluefin and GTs, which have been recorded in Hawaii especially, and we have some preliminary data to show this as well. So lots of new questions to ask. I can't name every name on here, but there were a ton of people that helped with this work, so I'm super grateful for the citizen scientists and sport fishers that helped us out collecting these samples, co-authors and collaborators on this work, a plethora of funding sources, and thank you all for listening. I'll just end with this slide up because there's a lot of papers that came out of this, especially the whole genome work that might be of interest. So feel free to read more or reach out to me for further questions. And if there's time, I'll take questions now. Thank you. Yeah, that's a really great question, and that's like would be the next step. Yeah, so we published this work. Unfortunately, one of my main collaborators that was working in Hawaii passed away suddenly, so we're still working on that. And there's some other information that we're gathering about like sex determination, but that's the next step, and it's sort of like I think what we're all grappling with in many of our fields of like, okay, where do we step in to like integrate into the management part? And there's definitely space to like build additional collaborations and connections in Hawaii. I haven't, you know, the Division of Natural Resources in Hawaii was aware of this work because they helped us get samples, but I haven't followed up a lot or since then to really like investigate what's happening. So yeah, great question. Yeah.